much for the presentation. I wonder if I could please ask a first question.
Yes. Well, it depends what it is.
Okay. Well,
Go on.
thank you. You referred to the sensations that you would receive when Irena was moving her hand as a pulse, 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 and several other times in your talk you referred to the neural sensations or the cognitive sensations you got sort of as like pulse, pulse, pulse. But I wonder if you could use those primitive sound wave depressions and other aspects of the human language to really give us what exactly did you really feel? I mean, say more than pulse, pulse, pulse. Tell us what it felt like.
I mean, it is quite a technical thing. I mean, essentially, we opened up a new passage to my brain via the nerve fibers, but my brain had to be retrained to pick up signals via this passage that we'd got. And it really was, I mean, for those of you that 80 microamps at 40 to 50 volts. Sorry, this is highly technical in a sense, and rather than an emotive one. At about that current input, my brain was able to distinguish pulses. Now, when we first did it, so essentially I'm there with a blindfold on, my researcher presses a button. Sometimes it injects a current, sometimes it doesn't, but I don't know. And I'm trying to say, yes, it does. So I'm concentrating, trying to focus on that. After about 2 weeks of doing this, my brain was able to get about 70% accuracy, which is not wonderful, but it's a start. And what it felt like was, it was as though there was a charge running up my index finger. So that's the sense my brain was receiving a pulse, and the sense it made of it was there's a charge running down my finger.
So like for those of us who wouldn't ordinarily have that, is it like when you put your finger in an electric outlet, but not all the way to getting a serious shock, you just feel kind of some electricity?
Feel electric, but it was more of a tickle. I liken it when the implant went in, for the first few days, as it settled down, as the nerve fibers settled down, I was getting sort of just ticklings, as you know, like a bit small. You knew they were electricity, but very, very light. This was stronger than that, but it wasn't painful in any way.
Sort of like, okay. And then how about the radar experiment?
Yeah, I mean, then when we did the ultrasonics, because I knew we were doing the ultrasonics, it was the same signals that we were using, but my brain didn't think, oh, that's my finger hurt. It just was able to make that link. There's an object the distance away, because it knew what was happening. And it did that very quickly. It wasn't like I had to have 3 hours moving around before I was able to do it.
Was it that same electric tickle that your brain felt?
Well, it was just it was just picking up a pulse. It was the same actual pulse of electricity, or the frequency was changing, obviously, but it was the same 80 microamps. And
how did you subjectively interpret that?
I interpret it that there's an object that is that far away. And it was the same when my wife sent me a signal. That's my wife sending me a signal. So I think first of all, your brain is very plastic. It can adapt and start picking up new signals, but it does make the best guess at what those signals are. Maybe it can get them wrong, but it's able to, oh yeah, that's what we do now, that's what it is. And when it's obvious, I guess we do make mistakes. Our vision often overrides. In the ultrasonic case, I had the blindfold on, so.
Next question, please.
Thank you. Marina Brockway. Dr. Warwick, I commend your dedication to medicine. You stand among the people who did catheterization on themselves to prove the value of that, in my appreciation to you. The question that I have is, as far as I understand, median nerve has also autonomic nerve connection. At least from what I know, is there is an acupuncture point right near median nerve that is implicated in treating ischemia, or at least relieving ischemia. And my question is, did you feel any autonomic response when the nerve was activated?
I have decided, not really. Not that I can report on, no. I mean, I know in terms of acupuncture and things like that, there's obviously an awful lot that we can learn about how we could link the two things together. I think now people are a little bit more open to the links between the two and the potential medical benefits and so on. But in my case, I can't claim that there was any other. I mean, I was looking at it as I've just described in a very technical way, I'm afraid, and just reporting what I could report. So I'd like to say yes, but.
Next question, please. Please introduce yourself and affiliation.
Jun Lee, I'm from Kansas State University, but I work on develop materials to improve the brain, the neuroelectric interfaces. But that work was done while I was at NASA Ames Research Center. And thanks for this exciting presentation. It really, really excited me. Okay, I have two questions. First, with that little robot you controlled with neural cells, and how long do you keep them there? And over that course, while it's learning, did you see any change in terms of the morphology, the number of neurons, and do they develop or they decrease?
Yeah, I mean, the answering those questions through, typically we had put it in for about 1 hour. We've got about 25 brains on the go at any one time, one robot in the pen, and we will put the brain in the robot for about an hour. But what one of the researchers is supposed to be doing now is putting the powered floor that we saw in the original clip, putting that for those little robots, because what we're looking to do is a 24/7 activity. So we can keep the culture. It's actually kept in an incubator with a Bluetooth connection to the robot. So we can keep that in appropriate conditions. And what we're looking to do, this is now a futuristic bit, is to have it operating in the robot 24/7 so we can witness what's going on in the culture. In terms of changes, yes, we do see strengthenings of pathways in a very big way, but just over the habits, over a week on a week on a week, when we look at snapshots. Now, how was this brain 3 or 4 days ago? It was, oh yeah, look, we can see this pathway now. Of course, we can't ourselves say, yes, this is definitely what this pathway means, but you can hazard a guess. I mean, interestingly, it does happen that Ray was talking to me yesterday about hierarchical nature of the human brain. Well, what we do see in our little culture brains is that sort of thing occurring, that some of the neurons, more by chance, I guess, tend to be sensory neurons, others tend to be motor neurons, and yet others seem to be in a sort of controlling position and rerouting, and as though they're sitting back and saying, hey, you've got to do this, you've got to do that. So the brain within a brain type of thing, and we can witness that just from a habit point of view. But we're clearly learning. Chemically, you can apply chemicals to increase the rate of neuron connection or to decrease it, but to apply signals, electrical signals that in some way are meaningful to the brain is difficult to know what's good, other than experience. You say, yes, it seems to learn this better, and if we apply these signals, it does the same thing, so maybe that is a reward or positive function to the brain.
Yeah. Second question is regarding that Utah array you implant in your arm. Is any data compare with the implantation people use on the cortex? And also, you mentioned you keep there for 3 months. And do you see any degradation? Glial cell may kind of encapsulate it. And,
oh, yeah, it's a good. I have to say, first of all, no. I mean, it was the Utah array, which was later the company Cyberkinetics, and we sort of beat them at it, and I don't know that pleased them all that much. We'd sworn to them that we were only going to do animal studies, and then we went ahead and shoved it in me. They then used it in Matt Nagel, the guy. He subsequently died. but only for monitoring. They didn't actually apply signals. The only feedback there was through visual. They were just monitoring the signals, which was a great shame. But we haven't been able to compare our data with their data. In terms of how the array operated, on the positive side, what we were able to report, when it was removed, fibrous tissue had grown round the array, but it actually acted in a positive way. It pulled it in place, made the connection firmer, and acted as a sort of a bubble wrap, a mechanical protection. And certainly Philip Kennedy, with his from Atlanta, his work, he's described his implants as exactly the same thing. We compared results, and we seem to get the same, what seems to be a natural body response to encapsulate, actually it sort of encapsulates pulling the thing into the neural pathways, so it can work in a positive way. And that's what we witnessed there. So it's far from trying to push it out of the body. It's as though, you know, the nervous system and the brain, it's as though it's a different thing altogether to the body. And so the defensive mechanism is pushing it into the brain rather than pushing it out of the body.
Bob Sklobosky, Computational Diagnostics in Pittsburgh. This is just a lot of fun stuff. I just love it. I have one comment, and what was going to be a question, which has probably turned into a comment because of the previous question. But the comment is on the visual experiment you did with the sonar and stimulating the median nerve electrically. And I think that's a very good example of plasticity of the nervous system, namely the nervous system being reorganized. And there was a beautiful series of papers published by Paul Baccarata in the 60s where he took blind people and with pressure stimulators attached to their back was able to recreate their visual experiences. And I think this experiment with a different approach to it does exactly the same thing.
Yes, it fits in nicely.
Yeah, it fits in greatly with that. And my question, and I guess I'm not sure if it's a question or a comment at this point, goes back to the neural mass problem that you have where you grow on the the neurons in the bath and then use those to control the robot. And the way I pictured that in my mind was you had a neural mass, and essentially you're performing a conditioning experiment on it. You've got the feedback coming from the robot to one end of the neural mass, and somehow you've created an effector output from the other end. And then you go through this conditioning paradigm where the robot hits the wall and you turn something on. And
Yep.
Exactly. So it's a classic feedback kind of thing.
Oh, exactly, yeah.
And what I was picturing in my mind, and I guess this is really my question was, and you've kind of answered it, was the change in the synaptic strengths, and you would predict a Hebbian synaptic property occurring that you should be able to see some synapses, and probably electrically, I would presume you had some electrodes sticking in this mass to also see what was happening at different junctions and things.
We will be. I mean, at the moment, because it's a flat array that it's growing on, so we haven't actually started prodding anything in it. But we have been getting the time sequencing so that we can, whilst the robot is moving around in the corral, what we're trying to get going is a video sequence of the brain, what it's doing in terms of firing activity.
So use fluorescence or something?
Yeah, that's right, in order to show what's happening as the robot gets nearer to the wall. Oh, look, this area of the brain. Yeah. I mean, it really is trying to study some of the basics of memory. As to what they are.
Yeah, it's learning memory, the conditioning.
Yeah.
Just a powerful
paradigm. But I'm excited. I ask you, am I allowed to ask questions? I mean,
I'm not allowed to answer.
We have. Okay, well, this is maybe to everybody. We've used rat neurons, which rats are not popular guys, so there's not a problem with that. But we have human neurons now, and we're culturing the neurons. To me, great, because from a medical point of view, this would have much more meaning than in terms of the rat neurons. But the whole thought of what the media does with it, is a robot with a human brain, which is what they're going to say. I mean, how do people feel here? Is everyone, go and do it? Or, well, this is any advice, or I don't know.
Well, the media will have fun with it, I can tell you that. Actually, there was I don't know if people remember this or not, but there was a fella in Cleveland named Robert White, and he was a neurosurgeon, and his goal was to transplant brains.
Yes. Yeah.
And he did a series of experiments where he would take a living dog and decapitate another dog and attach the second dog's head to the body of the first dog. And so while he was doing these experiments, which kind of actually worked, I mean, in some ways the second head took and didn't quite function right, but was kind of interesting. But the popular press got hold of this stuff, and they had these incredible articles showing, you know, the photograph of a dog with two heads, and, you know, you could see it in the National Enquirer. So you could expect that kind of stuff to come
so
forward.
to behead me. I think.
Thanks a lot.
Thank you.
Eric Jacobson, University of Illinois, and perhaps this relates to your question. About a month ago, I went out to the video store for an evening's entertainment for myself and my wife and brought back sleepers, which you're perhaps familiar with. So it seems as though you've done the core work for inventing the orgasmatron. And so it may be that aspect of it, when you go to using human nerves, might perhaps be emphasized.
That'd be more popular. I have to, just as you're going, 3-4 years ago, I spent 3 months as a Senior Beckman Fellow at Champaign, University of Illinois, and it was whilst my time there that the work on the rat neurons, that work started, really. That is my time spent there getting me into this work. So thank you for the University of Illinois through yourselves for inspiring that work. And if we do come up with the orgasmatron, then Illinois will be to blame for it completely. Fine. Yes, 10% of all the orgasmatron takings go to Illinois. That's the state of Illinois.
Just as long as it doesn't buy a senator's seat.
Oh yeah, that's much cheaper than that.
My name's Linda McDonald Glenn. I'm an attorney and a bioethicist. I should say recovered attorney and a bioethicist. And I'm intrigued with the ethical questions, and certainly have given much thought to how to deal with this in the press. And there's a lot of questions. Yeah. Do people understand that this is not sentience, or is it? So we get into the issues that we need to talk to the neurologist, the neuroresearchers with, because certainly there's an understanding, a concern about pleasure and/or pain that sentient beings experience, which is one of the reasons they shut down that dog experiment. And I was wondering if you had have you heard of the sentience quotient that was developed by Bob Freitas? I mean, there's some issues with it, but some problems with it, but it's at least
I'll look.
And
I have to say on this point, when we'd just done this research, which is still fairly recent, only to the end of last year, for the rat brain, one of the biology assistant professors in my university came up to me while I was just walking across campus and said, "Hey, I've been thinking about this. Do you realize that your rat brain robot, for all the definitions we have in biology, is alive?" So
right.
So
it's alive, but there's a different. But a cell is
a
cell is alive too.
Yeah, that's right. Yeah.
I could talk to you for hours about this, obviously. So maybe I can talk to you about this later.
Oh yes. Yeah.
Okay, thank you.
Look forward to it.
Hi, Melanie Swan, MS Futures Group. I'm just wondering what determined the 3-month time frame for the project.
Yes, well, initially I think it was going to be 2 months, because it actually, when you're doing that, I was like a walking laboratory, so took up quite a bit, and all we were doing was that, so I really had no time off, because you've got this thing in your arm, so you want to use all the time you can to experiment. But we still had things we wanted to do at the end of 2 months, like the experiment with my wife we hadn't done by that time, so we continued it for a bit longer. Although the electrodes that were implanted seemed to be perfectly all right, we checked them afterwards, there didn't seem to be any material disappeared or anything like that. Where the wires came out of my arm, they were dying off. They were being broken, partly because I went in every week. The nurse just checked if there was any infection, and so they were moving around. And I think we ended up, at the end of the 3 months, we had 3 connections left. So all the other wires, and it looked as though it was where the wires came out of my arm. I mean, I don't think that it was designed to have that much moving around, but that was a key factor, that at some point next week maybe there wouldn't be any connections left, and we had to book up about 2 weeks in advance for the surgery for the neurosurgeon. So we guessed in 2 weeks' time, and we were fortunate we still had some connections, but not many left. But it really, I think the overriding factor was I was absolutely exhausted at the end of the 3 months because of doing this experiment in the lab every day, doing that sort of stuff. So that was a... Yeah. Oh yeah, they were doing all the work and I was having all the fun, but thank you.
Well, it's exactly 12:20. I'd like to thank our keynote speaker for an absolutely fascinating talk. I think we were talking about whether the device is alive or not. The reassuring thing that people in the field of deciding when, trying to look for life, is there isn't any definition. There's a series of characteristics that many of which they have. Oh, yeah. Well, forest fires have some characteristic of life as well. And but again, this talk has just stimulated our imagination, and it also reminds us of trying to drink from a fire hose. There's so much information that's coming in at one time, but we really appreciate your giving us this very stimulating talk. Thank you. So we're off to lunch, and we're pretty much on time, so we'll come back as planned. That's great. What's the step? Yeah.